Hey guys, so yesterday we learned all about the different parts of the cell. Today we're going to be really focusing in on the cell membrane, including its structure, as well as some of its different functions. Specifically, we're going to be focusing a lot on the different ways that the cell membrane helps transport materials into and out of the cell. So this lesson is the cell membrane and transport. We're going to be focusing on, like I said, both the structure of the cell membrane as well as its functions specifically with transport. First, you need to know that the cell is considered an open system, meaning that both matter and energy can move into and out of the cell. This is important because the cell has to get nutrients into it, waste out of it. It has to be able to move ATP, which is a usable form of energy, as well as chemical energy into and out of it. And all of that is necessary to support different life processes, different metabolism of chemicals, and to just maintain homeostasis, which is a stable internal environment. The cell membrane plays a really important role in maintaining this open system. It essentially controls what molecules can move into and out of the cell. The cell membrane is called a phospholipid bilayer because it is made of two layers of phospholipids. This is what you're seeing at the bottom here. So these, kind of like the red ball with the two little yellow things dangling off of it, that is a single phospholipid. And then we have two layers that face one another. That is the phospholipid bilayer. Each phospholipid has a hydrophilic head, which means that the head likes to be in water, and a hydrophobic tail, meaning that the tail does not like to be in water. And so because of this, the heads face outside the cell membrane, so either to the inside of the cell or to the outside of the cell, both where there's aqueous environments. And then the tails hide between the two layers of phospholipids, like in the middle of that bilayer where there is no water. There is other structures in the cell membrane, even though it is primarily composed of phospholipids. There is also cholesterol molecules, which just help provide structural support or strength to the cell membrane. There's also protein channels, which are also called transport proteins and also called pores. They essentially look like a paper towel roll that goes through the cell membrane, and it is used to transport some materials. There's also peripheral proteins, which are just on the outside of the cell membrane, and they hold different cells together, and they're also used in cell-to-cell communication. Then there's carbohydrates. Those are also on the outside of the cell membrane. They act as attachment sites for different molecules. And then lastly, there's something called glycolipids, which is a structure also on the outside of the cell membrane that's used to identify the cell. So essentially, like cells of your immune system can come up to different cells of your body and recognize that they are cells of your body by attaching to the glycolipids. All of these structures can move around within the cell membrane. They kind of float past each other. And because of that, the cell membrane is referred to as a fluid mosaic model. Fluid meaning that the parts aren't stuck, they can flow past one another, and mosaic meaning that it's made up of different pieces. There's a couple different jobs of the cell membrane. One is to just limit the size of the cell. Another is to just create a boundary between the intracellular or the inside of the cell area and the extracellular area, which is outside of the cell. The cell membrane is selectively permeable, which means it selects or chooses what can pass through it and what can't. It's also referred to as semi-permeable, which just means that some things can pass through it and some can't. And then it's also important for both recognition and communication between cells. When we talk about cell transport, we're referring to the movement of molecules either into or out of the cell. And anytime molecules do that, they have to go through the cell membrane. Only some molecules are able to actually do that. There are two different kind of main categories of cell transport. The first is passive transport, and the second is active transport. A concentration gradient is one of the ways that we differentiate between passive and active transport. If molecules are moving with the concentration gradient, they can do that passively or without energy. If they are trying to move against the concentration gradient, it would be like trying to fight against the flow of a river, and that would require energy. So we call that active transport. Essentially, a concentration gradient is just a change in concentration from an area of high concentration to an area of low concentration. And concentration is just how many molecules there are in a given amount of liquid. It's kind of like density, but for liquids. So if you have a higher concentration, it means that there's more molecules dissolved in a smaller amount of liquid. So if a molecule is going from high to low concentration, we say that it is moving with the concentration gradient, and that will just happen naturally or passively. If the molecules are moving from an area of low concentration to an area of high concentration, then they're going against the concentration gradient, and that is going to require energy. It will be active. There are 3 different types of passive transport, which is again transport that does not require energy because it is moving with the concentration gradient. Those 3 different types are diffusion, facilitated diffusion, and osmosis. So diffusion is just when molecules naturally move from high to low concentration across a cell membrane. Once enough molecules move across the cell membrane, you will eventually have an equal concentration on both sides. At that point, we say that that cell has reached equilibrium. You will still have molecules moving through the cell membrane, but you will have an equal amount moving in as what moves out. So the concentration on either side doesn't change. Some of the factors that affect the speed of diffusion include the size of the concentration gradient. So if there is a bigger difference between the high and low concentration area, molecules will diffuse faster. Also, the smaller a molecule is, the faster it can diffuse. The hotter the environment that the molecule is in, the faster it will move, and the shorter the distance, the faster molecules can diffuse. And so this is just kind of showing what diffusion is. This, on the left-hand side here, would be a high concentration area. On the other side of that semi-permeable membrane is a low concentration area. Over time, molecules would diffuse from the high concentration area into the low concentration area until it reaches equilibrium, where you have an equal concentration on both sides. At that point, you would still have some molecules moving from the left to the right and some molecules moving from the right to the left, just balancing each other out. Facilitated diffusion is the same kind of process. You still have molecules just naturally moving from an area of high concentration to an area of low concentration, but facilitated diffusion involves molecules that cannot just go directly through the cell membrane. Something needs to help or facilitate their diffusion. So in this case, molecules will diffuse through a transport protein, also called a protein channel, in order to help them get through the membrane. The reason this would happen is if molecules are just too big to fit through the cell membrane, that they might have to go through one of those transport proteins instead. Also, if you have a hydrophilic molecule. Hydrophilic molecules only like to be in water, and the middle of the phospholipid bilayer, where all those hydrophobic tails are, there's no water there. And so hydrophilic molecules are not able to go through that middle area of the cell membrane. So instead, they'll go through a pore. The entire inside of the pore or transport protein is all water, so it can go through there more easily. So this is just kind of comparing what this would look like. Again, with just normal diffusion, those molecules can go from high to low concentration straight through that phospholipid bilayer of the cell membrane. But if you have molecules that are either too big or that are hydrophilic, they will have to go through one of these protein channels or transport proteins instead. That is facilitated diffusion. They're still going from high to low concentration though. And then the last type, which is the most different type, is osmosis. And this is where you have the diffusion of water from an area of low concentration of dissolved solids to an area of high concentration of dissolved solids. This one might seem like it's going against the concentration gradient because we are going from low to high concentration of dissolved solids. But remember, it's not solids that are moving here, it's water. So water is going from areas where there's lots of water and it's really dilute to an area where there's not a lot of water. So it's going to try and dilute an area of high concentration of dissolved stuff. It's hoping to essentially balance out how dilute the two different areas are. So in this case, the semi-permeable membrane is not allowing for the movement of the dissolved stuff. So the red particles can't move. We have more of the red particles on the right-hand side, and so the water is going to move over there until the concentration on both sides is equal. They're equally dilute. So water always moves towards where there's more things for it to dissolve. Tonicity is related to osmosis. Essentially, it has to do with the amount of water versus dissolved stuff inside of a cell. So if a cell is hypertonic, that means that it has a lot of dissolved solids in it relative to the outside, and therefore water will flow into it. That should say hypotonic. Yeah, hypotonic. Water will flow into it until it becomes turgid and could potentially eventually burst because of how much water is going into it. If the cell is hypertonic, that means it doesn't have very much dissolved solids, so water will flow out of it. This causes the cell to become really limp as it loses that turgidity. And then if a cell is isotonic, that means that the inside and outside have equal concentrations of dissolved solids. So this is kind of what it would look like. So in a hypotonic case, you have more water flowing in than what is flowing out because there's a higher concentration of dissolved solids on the inside of the cell. And so the cell will swell and become turgid or stiff. In the hypertonic situation, you have more water that is leaving compared to what's going in because the concentration of dissolved solids is higher in the solution. And so you get a cell that shrinks and becomes really limp. And then in the isotonic situation, you have an equal amount of water going in and out. So the cell is not going to change shape. So those are the three different types of passive transport. Then we get into active transport, which is the type of transport that requires energy because molecules are moving against the concentration gradient from a low to a high concentration area. There are four main types. We have uniport, antiport, and symport transport, as well as vesicle transport. So uniport is when molecules are pumped across the cell membrane through a protein channel. Anytime I say the word pumped, that means that it is active transport. It requires energy to pump something. It's not just flowing along the concentration gradient. With uniport, we have one type of molecule that is able to move one direction across the protein channel. And obviously, because this is active transport, it will be moving against the concentration gradient. That is only slightly different from antiport. With antiport, we still have molecules that are pumped across the cell membrane through a protein channel, still requiring energy because we're moving against the concentration gradient. The only difference is that now we have two different types of molecules that are moving in opposite directions of one another. An example of this that you need to know is the sodium-potassium pump, where sodium ions are pumped outside the cell and potassium ions are pumped into the cell, both against their individual concentration gradients. And then the last type of port transport, again, molecules pumped across the cell membrane through a protein channel against their concentration gradient, so requiring energy. The difference this time is that we once again have two different types of molecules, but they're moving in the same direction as each other. So either both out of the cell or both into the cell. So this is just comparing those different examples. So again, uniport is going through the protein channel in one direction, so one molecule, one direction. Antiport, two different molecules in opposite directions, and symport, two different molecules in the same direction. And they are all moving against the concentration gradient and therefore requiring energy. And then the last type of active transport is transport involving vesicles. So vesicles are small membranes that stuff can be packaged inside of. This can either move things into the cell or out of the cell. If vesicles are moving molecules into the cell, it's called endocytosis. If vesicles are moving things out of the cell, it's called exocytosis. With endocytosis, there are three different types. There's phagocytosis, which moves large molecules into the cell, pinocytosis, which moves liquids into the cell, and receptor-mediated endocytosis, where you have little receptors that are on the outside of the cell membrane, and molecules that are on the floating around outside the cell will like bind to the receptor and then get pulled inside into a vesicle. The exocytosis type is the type that actually starts at the Golgi bodies inside the cell, where the Golgi bodies packages fats and proteins and stuff inside of vesicles and then sends them off towards the cell membrane. So these are the different types of endocytosis. So this pinkish area is the inside of the cell, the bluish area is outside of the cell. And so the cell membrane would come up and fold around a molecule that is floating around outside the cell and then grab it. These two sides of the cell membrane right here would touch and then kind of separate. This top area right here would reform the outside of the cell, and then this circle part right here forms the vesicle. So that would be our phagocytosis. It would work the exact same way, just with some liquid in pinocytosis. Again, this part would just end up going straight across to form the cell membrane again, and this part down here would just kind of pinch off to form the vesicle. And then these are the little receptors that are on the outside of some cells. They will bind to molecules that are floating by, and then once again, this will kind of pinch off to form this vesicle. So yeah, this is just kind of slowly showing the process in a few more steps. So you have the cell membrane, it buckles downward to form this little pouch, and then it just keeps coming downwards until this part just pinches off to become the vesicle, and the cell membrane stays nice and flat. This is the one that would be a vesicle coming from the Golgi bodies, and it's going to go up and bind to the cell membrane and then release its contents outside of the cell. The last thing I do want to quickly touch on that's not related to transport, but it is related to the function of a cell membrane, limiting cell size. If cells get too big, if their surface area to volume ratio becomes too small, they don't function as efficiently. So the smaller the cell, the higher its surface area to volume ratio. It essentially means you have a lot of surface area for a small volume. This is why like crushed ice will melt faster than a block of ice, because it has a lot of surface area for a small volume. It's the same reason like shredded cheese melts better than a hunk of or slices of cheese. And so in order for molecules to be transported efficiently through a cell, the cell needs to be fairly small. And so that's one of the jobs of the cell membrane, is to make sure that the cell doesn't get too big. Okay, so this is just a little flow chart to kind of summarize these different types of transport. So on the one side, we have the different types of passive transport, which includes diffusion, facilitated diffusion, and osmosis. Diffusion is when you go from high to low concentration through the cell membrane. Facilitated diffusion, also high to low concentration, but through a protein channel that is for either large or hydrophilic molecules. And then osmosis is when water is moving from an area, to an area high in concentration of dissolved solids. It's trying to dilute those dissolved solids. For all of those different types of passive transport, there is no energy needed. On the other side, we have active transport, which I've broken down into the ports and the vesicles. For the ports, those are ones that are being pumped through a transport protein in the cell membrane against the concentration gradient. So with uniport, you have one molecule being pumped one direction. Antiport, two molecules pumped in opposite directions. That is our sodium-potassium pump. And then symport is two molecules being pumped in the same direction. Then we have the vesicle transport, which is also active, also requiring energy. Exocytosis is when the things are moving out of the cell. Endocytosis is when they are moving into the cell. That could be either phagocytosis, which moves large molecules into the cell, pinocytosis, which is liquid moving into the cell, and then receptor-mediated endocytosis, which is when molecules will bind to receptors in order to move into the cell. All of these ports and vesicle types of transports require energy. Okay, so that is it for today's lesson. Let me know if you have any questions.